Ever wonder if we could edit life itself, like a document on a computer? Welcome to the world of genetic engineering, where science fiction is rapidly becoming science fact. We're talking about technologies like CRISPR, the possibility of designer babies, and the deep ethical questions that come with rewriting the very code of life. It's a field filled with mind-blowing discoveries and complex challenges. Today we're diving deep into this fascinating universe. We'll count down the 10 most stunning facts about genetic engineering, tracing its incredible journey from a simple idea to a world-changing technology. Are you ready to have your mind blown? Let's get started. Number 10. The journey begins not in a modern lab, but with a humble garden pea. Back in the 1860s, a monk named Gregor Mendel meticulously crossbred pea plants, discovering the fundamental laws of heredity. He figured out that traits are passed down in predictable patterns through what he called factors, what we now know as genes. This was the first crucial step, the foundational insight that made everything else possible. Without Mendel's peas, the entire concept of editing genes would have remained pure fantasy. Number 9. Fast forward to the early 20th century. Scientists were racing to understand what these genes were actually made of. In 1953, James Watson and Francis Crick, with crucial contributions from Rosalind Franklin, finally cracked the code. They unveiled the iconic double helix structure of DNA, the molecule that carries our genetic instructions. This discovery was like finding the alphabet of life. For the first time, we could see the blueprint that makes us who we are. Number 8. Knowing the structure of DNA was one thing, but being able to manipulate it was another. The breakthrough came in 1973 when Herbert Boyer and Stanley Cohen achieved the seemingly impossible. They successfully cut a piece of DNA from one bacterium and pasted it into another, creating the world's first genetically modified organism. This recombinant DNA technology was the birth of modern genetic engineering, proving that we could actively edit the book of life, not just read it. Number 7. With the ability to edit genes, the world saw its first major application in medicine. In 1982, scientists used genetic engineering to program bacteria to produce human insulin. Before this, insulin for treating diabetes was sourced from animals, which was expensive and could cause allergic reactions. This new genetically engineered insulin was identical to what humans produce, revolutionizing diabetes treatment and saving countless lives. It was the first tangible proof of genetic engineering's power to heal. Number 6. The next frontier was agriculture. In 1994, the first genetically modified food hit the supermarket shelves, the Flavor Saveur tomato. It was engineered to ripen on the vine for longer without becoming soft, promising better flavor and a longer shelf life. While the Flavor Saveur itself wasn't a huge commercial success, it opened the floodgates for genetically modified crops. It sparked a global conversation about the future of our food supply and the role of science in what we eat. Number 5. The idea of cloning, once just a sci-fi trope, became a stunning reality in 1996 with the birth of Dolly the sheep. Dolly was the first mammal cloned from an adult cell, a feat many scientists believed was impossible. The process involved taking a cell from an adult sheep and fusing it with an unfertilized egg that had its nucleus removed. Dolly's existence demonstrated that the genetic code in a mature cell could be reset to create a whole new, genetically identical organism. Number 4. As we entered the new millennium, a monumental scientific quest reached its conclusion. In 2003, the Human Genome Project was completed, successfully mapping the entire sequence of human DNA. This colossal international effort gave us the first complete instruction book for a human being, identifying all 20,000 plus genes. This map has become an invaluable resource for understanding and treating genetic diseases, from cystic fibrosis to Huntington's disease. Number 3. The next leap forward was less about reading the code and more about editing it with unprecedented precision. While earlier methods were clunky, a revolutionary new tool emerged from an unexpected source: bacterial immune systems. Number 2. With the power of CRISPR, the once hypothetical idea of designer babies entered the realm of possibility, sparking intense ethical debate. In 2018, this debate became a shocking reality when a scientist in China claimed to have created the world's first genetically edited babies. He used CRISPR to try to make them resistant to HIV. The announcement sent shockwaves through the scientific community, leading to widespread condemnation and urgent calls for global regulations on editing the human germline, changes that can be passed down to future generations. And now, for the most stunning fact of all. Number 1. Today, genetic engineering is moving beyond the lab and into the clinic, offering new hope for previously incurable diseases. We are now seeing the first CRISPR-based therapies approved for human use. In late 2023, regulatory agencies in the UK and the US gave the green light to a treatment for sickle cell anemia, a painful and life-threatening genetic blood disorder. This therapy edits a patient's own stem cells to correct the faulty gene, marking the beginning of a new era in medicine where we can fix diseases at their genetic source. From Mendel's peas to curing genetic diseases, the journey has been nothing short of extraordinary. What a ride through the history and future of genetic engineering. It's a field that holds incredible promise, but also requires our careful consideration and wisdom. What do you think is the most exciting or concerning part of this technology? Let me know in the comments below. If you found this journey as fascinating as I did, please give this video a thumbs up and subscribe for more deep dives into the science that is shaping our world. Thanks for watching, and I'll see you in the next video.